Good evening, everybody. Thank you for joining us tonight. My name is Michelle Lorenz, and I'm with Everything ALS, a 501c3 that's here to help patients connect the care all the way through the cure. And our 501c3 was founded to be an asset for patients. This presentation tonight is to give you an overview of about ALS. So it can be for people that are newly diagnosed, just to give you some more background so that you can have a dialogue with your clinicians, or it could be for somebody who's a caregiver or a family member who just wants to know a little bit more about ALS. So we hope this brings value to you. Feel free to reach out to us every Wednesday night. Every other Wednesday night, we have a meeting where we bring top clinicians and researchers and people in the ALS community to add assets for you. So we've had Dr. Mira Kukovic from Harvard. She's at Mass General running the Healy platform trials. Jeff Rothstein has been on, Ed Rapp, Google Euphonio, Team Gleason, all sorts of incredible people in the ALS community that are here to help support you. So every other Wednesday night, you can go to everythingals.org to get that information, register at the events tab, and then you will get email updates about who's going to be doing the presentation. So every Wednesday night, 5 Pacific, 8 Eastern. Now let's hold on and we'll do a screen share here and we will get going on what you all came to hear about tonight. And I will minimize myself. So as I mentioned, here at Everything ALS, we are working on the care to the cure. And tonight we're going to give you a little bit of an overview. So we're going to talk about the history of ALS. We'll talk about the anatomy of neuromuscular disorders, the diagnosis and prognosis of ALS, some of the epidemiology information. And then we're going to spend a lot of time talking about the genetics or familial form of ALS, which is 10% of the cases. Another 90% of the cases are environmentally caused. That's called sporadic or SALS. We'll spend a little bit of time talking about the pathology. We'll touch on the reversals that Dr. Richard Bedlock is studying at Duke University. He's going to be speaking to us in a few weeks, so we're just going to give you the highlights, and he's going to dive deep into that. And then we'll just finally close up with just a bit about nutrition. Again, we'll bring a nutritionist in on another one of our webinars to give you more information about how to optimize your nutrition as you're battling this disease. Now, let's start with the history of ALS. What we know about ALS is it was discovered by Dr. Jean-Martin Charcot 150 years ago. And in 150 years, we've not had one viable treatment. It's one of the reasons we are so passionate here about Everything ALS, trying to find resources for you. Now, Dr. Jean-Martin Charcot also discovered another neuromuscular disease called Charcot-Marie Tooth syndrome that impacts both the sensory and peripheral neurons as well. So he's really one of the forefathers. Fast forward, everybody started to become aware of ALS when Lou Gehrig came down with it in the 30s. So Lou Gehrig, a famous player for the New York Yankees. For those of you here in the United States, we obviously commonly call ALS Lou Gehrig's disease. Outside of the United States, it's often called Motor Neuron Disease. In the 1960s, Jerry Lewis had the Labor Day telethon that brought more awareness to a variety of different neuromuscular diseases. And so that was sponsored by the Muscular Dystrophy Association. He both fundraised and brought awareness. And then many of you know Stephen Hawking, who unfortunately just left us a couple years ago. Stephen Hawking is one of the world's most brilliant physicists and a long-term survivor with ALS. In this generation, we have a lot of different organizations that are bringing a lot of value to people with ALS. We wanted to give you a highlight of those. In the top left, the Strikeout ALS Organizations, founded by Pete Frates, who had ALS, played baseball up at BC, and he and his family have helped raise millions of dollars, hundreds of millions of dollars, to help fund research for ALS with that Ice Bucket Challenge. In the bottom left, Dottie Weir, who is a rugby player over in the UK, is doing something very similar over in his country. Next to him in the baseball cap is Eric Stevens with ACTS ALS. Eric and his wife Amanda have been critical in helping get legislation passed to give quicker access to different therapies. Next to him is Tim Green. You see Tim played there with the Falcons. His organization is Tackle ALS, more fundraising and awareness. Next to Tim is Brian Wallach with I Am ALS. Brian is a lawyer and worked in the political arena, and he and his organization, along with ACTS ALS, have been critical in helping get some of this legislation drafted that you're going to hear about in just a couple of weeks from some of our congressmen and senators. Next to Brian is Ed Rapp of Answer ALS, and Ed spoke on our webinar just last week. Ed is the chair of Answer ALS, and Answer ALS is doing some critical research, bringing together the top research minds around the world. And they have a study that they've put together of 1,000 different people with ALS, and it's called Omics. And that omics study is collecting all of the different data from your genome to the epigenome, the proteins, your metabolome on a whole bunch of different patients so that they can have this database that all sorts of researchers can access. So huge, huge asset. Then above Ed, you see Steve Gleason. He played with the New Orleans Saints, and Steve has really led the charge to bring technology and make technology more accessible to people with ALS. And then in the middle, you see our founder, Indu Navar, there in the red sweater. Indu's pictured there with her husband, Peter, and she founded Everything ALS after she lost her husband, Peter, in 2019 to this disease. And as they were going through their battle with this, despite the fact that they were entrepreneurs and leaders in the tech industry and had virtually unlimited resources to attack it, they realized that there wasn't a lot of data out there for people, that patients weren't being heard. And so one of her missions is to honor Peter's legacy by bringing the data to all sorts of researchers and cutting out all of the red tape to try to make this a quicker trip to understanding therapies and cures for people with ALS. So let's start with teaching you a little bit about anatomy and the neuromuscular system. We know that there are several different types of neuromuscular diseases that the Muscular Dystrophy Association talks about. You see them on the screen here. We're going to obviously focus on the motor neuron disease, of which ALS is one. But when you are getting diagnosed, these are the different types of diseases they're going to have to rule out. So you see the peripheral nerve diseases. Charcot-Marie-Tooth is one of those. Some of you have had to rule out myasthenia gravis. In muscular dystrophies like Duchenne's muscular dystrophy that impacts children. And then you can have mitochondrial diseases like Friedreich's ataxia. So all of these are the reasons that you need to get to a neurologist that specializes in ALS, somebody at one of the ALS Centers for Excellence you're going to hear about tonight. Now, as we talk about our nervous system, we know that our nervous system has hundreds of nerves and billions, with a B, billions of different neurons. And that's why it's so critical that we take care of those. Now, part of our nervous system is the central nervous system. That's what you see highlighted in the red. That's your brain and your spinal cord. This is where the analysis of information takes place. And then in the blue, we have the peripheral nervous system. These are your neurons and your glia that are outside the brain and spinal cord. And in your peripheral nervous system, we have two different types of neurons. We have sensory neurons, which bring the information up to your brain so that your brain can process it. And then motor neurons that take the information from your brain and send out signals to the rest of your muscles, your organs, and your glands. In ALS, it's your motor neurons that are affected, not your sensory neurons. Now, we wanted to give you an example of what that's like. So your sensory neurons will detect if the stove is hot. Your motor neurons will allow you to take your hand off the stove. Your sensory neurons will get the signal that you're thirsty. Your motor neurons will take that signal from the brain and allow you to pour yourself a glass of water. Your sensory neurons will bring the signal up to your brain so you can detect that your nose itches. Your motor neurons will send the signals out so that you can move your hand and scratch your nose. Your sensory neurons at the beach, they smell the salt in the air, they hear the waves crashing, you may feel the sand underneath your feet. And if that sand is hot, that message goes up to your brain and allows you to detect that. Your motor neurons are what enable you to get the signal down to your muscles so that you can run and get on a blanket. Your sensory neurons send a signal up to your brain that detects that you have some drool on your chin. Your motor neurons allow you to get the signal out to your hands to grab a washcloth and wipe your face. Your sensory neurons, they just watch and see that a car cuts you off in traffic, and they send the signal up to your brain. Your motor neurons are the ones that take the signal from your brain down to your feet so that you can slam on your brakes. In people with ALS, the sensory neurons continue to work. Their motor neurons are what have the problem. So let's talk a little bit about the different types of motor neurons. We have upper motor neurons and lower motor neurons. And a lot of people mistakenly believe if you have problems with bulbar syndrome or your hands, that means it's an upper motor neuron damage. Actually, in ALS, it's both upper and lower, and the damage to them manifests in a different way. So your upper motor neurons, they originate in your brain and your brainstem, and they control the impulses for your voluntary muscle activity. They also help with glutamate, a neurotransmitter that connects your upper to your lower motor neurons. Your lower motor neurons are what you see over here on the right. So the motor neurons come from your cervical, thoracic, or lumbar spinal cord area, and they go to these muscle fibers. And these spindles, these muscle fibers here, once they get that impulse, they contract, they flex, they move. And there's a little bit of difference in what we're going to hear about axons and dendrites. In your upper motor neurons, the axon is short and the dendrites are long. In your lower motor neurons, it has to go all the way from your spinal cord, perhaps down to your feet, to your hands. And so you're going to find longer axons and shorter dendrites. And we'll talk about axons and dendrites in just a second. Now, just different diseases of upper and lower motor neurons. When you're first diagnosed with ALS, this may be why you only get one of these diagnoses originally. And it's all the more reason, again, to get to a neurologist with lots of expertise in the area. So if you only are showing impact on your lower motor neurons, you may be diagnosed with SMA or PMA. You may have progressive bulbar palsy. If you're only showing impact on your upper motor neurons, they may diagnose you with primary lateral sclerosis or another one of the upper motor neuron diseases. The thing that distinguishes ALS is that it impacts both the upper and lower motor neurons. And so it's not only ALS, but you can get frontotemporal dementia that is also occurring simultaneously with ALS or motor neuron disease. Now, remember we talked about the axons and the dendrites earlier. So here's a picture of a neuron in the yellow, and the axon is this yellow, looks like the tail that extends all the way off that neuron. And again, in the lower motor neurons, that axon is going to be longer because it's got a longer way to travel to give the signals to your muscles in your legs and feet and arms, right? So let's talk about the different characteristics of these glial cells in this neuron. So here you have your neuron. We mentioned the yellow is the axon. These green things on the drawing that look like little toilet paper rolls, that's actually your myelin sheath. So your myelin sheath is the protective coating around your axon. Think of your myelin sheath just like the rubber cord on your cell phone charger. If that rubber cord gets damaged and the wires inside start fraying, you're not going to get a good signal to charge your phone. Same exact thing happens with your neurons. If the myelin breaks down or has damage around it, it can't protect your axons. And the myelin is heavily, heavily dependent upon healthy fats. So really, really critical. You'll hear about that a little bit later. The myelin sheath is formed by these things here called oligodendrocytes. And they are, again, really, really important to help you insulate your axon and protect them. Now, the microglia, they look like these little powder puffs here. Your microglia, think of them as the immune cells of your nervous system. They basically help you clean up all the debris and junk inside your nervous system, and they're incredibly reactive to inflammatory stimuli and toxins. So that could be things like Lyme disease, by heavy metals, by bacteria, by viruses. So anything that causes inflammation in your neurons, your microglia are going to be the ones that are impacted the most. Then you also have astrocytes. Your astrocytes right here, this looks like a spider with lots of tentacles off of it. An astrocyte is critical for neuronal repair, helping your neurons repair themselves, neurogenesis, new neurons, right? They help maintain a healthy working environment in your neuronal system. They help with communication, signal transmission, keeping oxidative stress down. You're going to hear a lot about oxidative stress tonight. And importantly, when you were developing, your astrocytes are also what helped form your blood-brain barrier, and your blood-brain barrier protects you from getting those toxins in your brain from your bloodstream. So it's a really kind of a SWAT defense system to protect your brain, your blood-brain barrier. Now, let's talk about ALS and where this came from. So ALS is known for its progressive neurodegeneration, meaning it slowly is degenerating or damaging your neurons. And that's both your upper and lower motor neurons. So that's really, again, important. It's the combined impact of upper and lower motor neurons. It affects your nerve cells that are in your brain, your spinal cord, your peripheral nervous system. And when your neurons die, your motor neurons die, the brain loses its ability to send the signals to those muscles, just like we talked about earlier with that beach example, right? Now, in ALS, unfortunately, it impacts your voluntary muscles. And as that progression occurs, people with ALS will eventually lose function of their limbs. They'll lose the ability to speak and swallow, and unfortunately, the ability to breathe, and eventually become totally paralyzed. So where did this name come from, amyotrophic lateral sclerosis? So if you look over here on the right, A means no or none, negative. Myo means muscle, and trophic means nourishment. So amyotrophic means you're not getting any nourishment to the muscle, meaning you're not getting those signals to your muscle. And this is an example of what happens. It's when you have a healthy neuron. This is your axon surrounded by the myelin sheath, and you come down here to the axon terminals. They look like roots on a tree, and those extend into your muscle. And when they get the signal, they send an electrical impulse to your muscle to contract, to fire, to do what muscles do, right? As your neuron dies, you see an example down here on the bottom. So you see less myelin sheath protecting the axon, and you see less of these tree roots. Again, those axon terminals, so you're not getting the same signal to your muscle. So even though your brain is sending the signal, it's not arriving there. And what happens is this muscle starts to atrophy. So you see a really healthy, robust muscle that's flexing up here. This muscle, when it doesn't get the signals, it starts to atrophy. And so the other thing that you'll hear us talking about tonight as well is what happens at the synapses. This is at the end of your axon terminals. And there's neurotransmitters that are communicated there as well. Glutamate's one of them that's definitely related to ALS. Now, we mentioned before, it's not getting signals to your voluntary muscles. Here on the screen, you see that we have involuntary muscles like your heart, your bowels, your bladder, your blood vessels, your stomach, your intestines. Those are the involuntary muscles that are not typically affected by ALS. Your voluntary muscles are the muscles most people are familiar with, your skeletal muscles, so your legs, your arms, your hands, your feet, but also you have bulbar muscles. So the muscles in your mouth and your throat, your tongue, your ability to chew, to swallow, all of those are impacted eventually. And then unfortunately, your breathing muscles as well. So ALS doesn't actually impact the lungs. You may get a pneumonia because you're not moving around and because you're getting a bacterial buildup in your lungs, but that's not specifically the muscles. The muscles are your intercostal muscles here around your rib cage, and they help you breathe. Both breathing in and breathing out are controlled by those intercostal muscles. And so when those muscles start losing the neurons that are innervating them, then you lose your ability to breathe as well. Now, we know that ALS has been called a rare disease, and part of that is because there's not a lot of people alive who have ALS at any one point in time. And part of that is because the prognosis for ALS, unfortunately, is 99.9% fatal. Every 90 minutes, somebody is diagnosed with ALS and somebody dies of ALS. The average lifespan is between 2 and 5 years. But it's not really a rare disease. What we know is that 1 in 300 chance of you or somebody getting ALS in your lifetime. And the survival time with ALS varies depending on the type of ALS you get. It can be the average, again, running between 2 and 5 years. That's about 65% of the people in that time frame that will unfortunately pass away. But there are people, 20% of the population lives between 5 and 10 years, and 10% more than 10 years, and 5% more than 20 years. One of our goals with Everything ALS is to help people get access to the research, to get access to the therapies, develop therapies, so that we can move people into those long-term survival categories. Now, the epidemiology of ALS, we know around the world, ALS is called Motor Neuron Disease. Here in the United States, again, ALS or Lou Gehrig's disease. The age of onset here in the US is between 55 and 65 years old. There's about 6,000 people who die annually from ALS. Now, what we know is the male to female ratio, in the earlier ages, it's about 3 men for every 2 women that are diagnosed, and men are affected at a younger age. Some of that's partly because men are probably exposed to more industrial toxins in the professions that they're around, but also because they may have had more concussions or some of those other sporadic exposures. But as we age, by the time men and women hit their mid-60s, the incidence of ALS is equal between men and women. We know that the incidence is similar between men and women if you have the genetic form, the FALS, familial form. If you have the sporadic form, men tend to have more of a spinal onset. Women tend to have more bulbar onset. Now, incidence and prevalence, you'll hear these terms interchanged. So incidence is how many people are diagnosed in a year. The prevalence is how many people are living with ALS. So we know in the United States, there's about 16,000 people that are living with ALS. Some researchers believe that it could be as high as 30,000 just because some of those cases have been undiagnosed or misdiagnosed as one of the upper motor neuron or lower motor neuron diseases. We know the ALS registry that comes from medical reporting that there's about 5 out of 100,000 people per capita that have this ALS. If you look exclusively at the Medicare data and the people that are receiving Medicare, that raises to almost 12 out of 100,000 people. Now, that's the prevalence of ALS in the United States. The incidence of ALS, as I mentioned, is about 6,000 people that die annually, about 6,000 people that are diagnosed annually. Really interesting, the incidence of ALS and MS, an autoimmune disorder that ultimately impacts your myelin sheath as well, the incidence of both of those is about the same. But unfortunately, the turnstile effect is what one ALS patient called it. Because ALS is fatal, there's not nearly as many ALS patients living for a long period of time like there are MS patients. And so again, one of the goals is to help extend that lifespan. And then you would find the prevalence of MS and ALS are almost exactly the same. If we look at motor neuron diseases around the world, we see there's about 330,000 cases, about 35,000 deaths annually. And unfortunately, about 5 per 100,000 people is the prevalence. One of the interesting things is the highest prevalence around the world is in the countries that have the highest socio-demographic index. So you look at the countries in the peach or red color, they have the higher prevalence of ALS. The countries in the blue, the lower prevalence. Now, we want to talk to you about providers because it's so incredibly, incredibly important to get yourself to an ALS Center of Excellence. So the ALS Association has gone around the country and done a validation to make sure that there are clinicians that know exactly what this is. They're going to be able to help diagnose you more quickly. They're going to have seen, instead of maybe 1 or 2 ALS patients in their life, they may have seen 10,000, like somebody like Jeff Rothstein at Johns Hopkins at the Packard Center there. So really important to get yourself to one of these locations. You can go to the ALS Association website and look these up. The difference between the gold and the silver treatment centers is the gold centers have the same level of clinicians, but they also have some research going on at those facilities. The silver badge on some of those organizations, the same level of expertise with the clinicians, but no research that's going on, no clinical trials. The other benefit of getting to an ALS Center of Excellence is you're going to see almost all of these different types of clinicians or practitioners, not only your neurologists, but healthcare professionals such as respiratory, physical, and occupational therapists, nutritionists and dietitians. You may have speech pathologists to help you retain some of your muscles in your bulbar region. The only thing that seems to be missing is having access to naturopaths and functional medicine doctors. And you'll hear later on when we talk to Dr. Bedlack's reversals and we talk about them, almost every one of those reversals was also seeing a functional medicine doctor that believes in the holistic approach, looking at the root causes of the diseases. They're going to do a lot of different types of testing. They're going to help you more with your diet and your supplements that can help your body's defense to ALS. Now, so what are some of the symptoms? For those of you that have ALS, you know these, but for family members or people that are newly diagnosed. So if you have lower limb onset, that's typically a lower motor neuron. That would be difficulty walking, clumsiness. Maybe you trip and fall. Maybe you have trouble dribbling a basketball or swinging a baseball bat for your kids' baseball practice. It could be difficulty holding a needle if you're doing sewing. Any of the things that deal with your hands or feet, your fine or gross motor skills. You could also have difficulty swallowing, difficulty chewing your food, your mouth gets tired. You may have muscle spasticity or hyperflexion in your muscles. And those are just some of the common symptoms. Now, if it's an upper motor neuron damage, you're going to see things like rigidity and stiffness, spasticity in those muscles. You're going to have decreased range of motion. You're not going to be able to lift your arm all the way back over to the top of your head as easily. You may have what's called a Babinski sign, which is like a hyperflexion of your toe and your foot. You may have pronator grip, which means you have your palm face up and it will slowly turn around and face so your palms face down without you intending to do it. Those are all signs of upper motor neuron damage. You don't typically have the muscle wasting as quickly when it's upper motor neuron damage. When it's lower motor neuron signs, you're going to have muscle weakness and wasting. You're going to have loss of your muscle tone, so flaccidity. You're going to have a lower amount of reflexes or absent reflexes. But one of the most common symptoms, in addition to the muscle weakness and weakness and wasting, are going to be fasciculations, which are muscle twitches that are going to be going on in muscles around your body, and different types of cramping. Now, ALS can have an onset in a variety of different limbs. It can start in your feet, in your hands. It can start in your bulbar region, and it varies depending on the type of sporadic or genetic onset disease that you have. What we do know is that it tends to be asymmetric. So if it starts in your right leg, it will move to your left leg and then typically moves to your hands and arms after that. So not always, but that's kind of the typical pattern, is it typically demonstrates on one limb, one side at a time. If you have bulbar symptoms, one of the things you may notice is difficulty swallowing. So this chart came from the ALS Association, from one of their speech pathologists. You'll find a lot more information on their website, but we wanted to give you an overview. So initially, you may just have troubles chewing or swallowing. Chewing a piece of gum may make your mouth sore. You may have more saliva and harder to swallow the saliva. Some of the food residue in your mouth, things that you haven't noticed before when you've been chewing. The pharyngeal stage, you're going to see food getting stuck in your throat. You may have trouble coughing or choking. Your tongue may be more difficult to move, may get more swollen. And you're having a reduced strength when you're coughing. You're not able to cough as effectively. The esophageal stage is what happens when you get towards the end of the bulbar onset. And this is when most people go on a feeding tube or a PEG. Just as a caution, if you do use a feeding tube or PEG, make sure you check out the formula so that it doesn't have a lot of really harmful things in it. We'll talk about that more in the nutrition phase. These are some of the common diagnostic tests that will be done by your neurologist to help not only diagnose you with ALS, but to rule out some of those other neuromuscular diseases we mentioned. We're hoping that this omics study that you see down here at the bottom that's coming out of Answer ALS will help give us some more biomarkers and diagnostic tools. There's a lot of research going on in blood research to find, and CSF research to find more biomarkers to help diagnose you earlier so we can get you into more clinical studies and therapies to help out. And when you're first diagnosed, your neurologist is going to check the signs in both your upper and lower motor neurons. And this is just some of the criteria that they will look at. So if you only have the signs of damage to your upper or your lower, they're going to say that's suspected ALS, but initially they're probably going to say you probably have one of those other neuromuscular diseases. As you move up the scale, if you have upper and lower motor neuron, or you have just in more than one region, you're going to find that this is probable ALS, and they're going to look to get an EMG to see where that innervation is. The denervation is, where you have less nerve activity going into your muscles. Now, if you don't have that EMG, but you have the symptoms in a couple areas, they're going to say it's possible, not probable. These gray boxes where it says suspected, possible, and possible, that's what happens when you don't have a clinical validation from some objective tests. So whether that's a genetic test here that helps detect that you have familial, or the EMG that says it's probable ALS, eventually you will have damage to both upper and lower motor neurons if you have ALS, and that's when you'll get a definitive diagnosis. Remember, we talked earlier about 16,000 people in the United States who are known to have ALS. Those are the people that have the definite probable diagnoses in the red. There are people in the gray boxes of suspected and possible ALS, and those aren't captured in that data, and it's why some researchers think there may be as many as 30,000 cases. Now, we want to encourage you again to see a naturopath or functional medicine doctor, because they're going to do some of these other tests that some of the traditional clinics may not do. We want to encourage everybody to get your entire genome sequence. Don't just check for the genes related to ALS. We'll talk about that in the genetic section of this presentation. Get a comprehensive stool profile. That means having your fecal sample run to see what your gut microbiome is. Again, we'll talk about that a little bit later. And then these blood, urine, and hair analysis, that's going to help you know things like your inflammatory markers. Now, your regular physician will measure a couple of inflammatory markers. There's a blood panel that measures over 200 different inflammatory markers, and inflammation is one of the biggest markers of ALS, so we want you to get that done. Check your amino acids, your organic acids, your citric acids. and so important to check your essential fatty acids, because your fatty acids are what form that myelin sheath that helps protect your axons. We want you to get your fungi and bacteria and viral load tested. Those are the toxins that can get through your blood-brain barrier, and the hypothesis is that they cause damage to your neurons. And you can do things to treat yourself to eliminate some of those toxins. Similarly, although it's a little bit more controversial, is the impact of Lyme's disease, tick-borne diseases, and heavy metals. But we want you to get tested for those, whether they cause ALS or whether they are a mimic. Certainly those are important to get treated. Any of the mycotoxins as well. So make sure you get to a functional medicine doctor, take them this list, and get these tests done so that you can help empower your body to get rid of some of the things that maybe, even if they didn't cause your ALS, they're helping speed the progression. Now, there's a way that you're going to track your progression of ALS. It's called an ALS FRS score. It's what your neurologist will use. And so there's 4 different categories. There's the category related to your bulbar region, speech, salivation, and swallowing, the category relating to your breathing, the respiratory category, the gross motor skills, which is your ability to walk, to run, to climb stairs, and your fine motor skills with your handwriting, using a pen to write, using a knife to cut your food, being able to button a shirt or zip your pants, use a toothbrush or a razor. Those are all measurements that they're going to evaluate. And so there's a place you can go called mdcalc.com. And this website will actually give you some great guidance. So they actually have the ALS FRS scores here, and you get between 0 and 4 points for every one of those criteria. So the top here, you see the fine motor skills. On the bottom, you see the clicks for the gross motor skills, and you can evaluate yourself and track this on a monthly basis. It's what your neurologist is going to look at on an annual basis when you visit them. Here's the ones for speech, and here's the one for respiration. So we also now want to give you some tools to teach you about genomics, because one of the things we've discovered is very few people are getting their genome sequenced when they have ALS, and there's a whole lot of reasons that we want to encourage you to not only get your genes tested for ALS, but your whole entire genome, all of your genetics. So what we know about the genetic form of ALS, it's called familial, or FALS. You may see that abbreviation, and it affects about 10% of the people that have ALS have the inherited form. And that means you inherited it from either one or both of your parents. And there's different genotypes. So the difference between a genotype and a phenotype, the genotypes are your genes, the phenotype are the symptoms. So you can have different genotypes. There's over 24 different genes that have mutations that cause ALS, but there's about 160 different genes that are associated with ALS. Alone, they don't cause ALS, but in combination with other factors, they contribute to ALS, and that's one of the reasons we want you to get your whole genome sequenced. We also want you to do that because some of those genes can be affected by the drugs you take, by the food you eat, the supplements you have, the water you drink, the saunas you do. That's called epigenetics. So there's about 80% of the genes in our body that we can impact how those genes are expressed. There's only about 20% of them that are hardwired. So if we can impact the expression of those other 80%, that's going to be helpful for you to know as well. Now, we're briefly going to talk about what's called modes of inheritance. So this means whether you get it from both parents or one parent. So what you see on the drawing here is 2 different chromosomes. And on those chromosomes, you have the genes. So in the drawing, the white dot are the genes that are not affected by ALS, and the blue dot is the gene that is affected by ALS. And what we know about ALS is that most types of inherited ALS are autosomal dominant, meaning you get them only from one parent. So if only one parent has the disease and they pass this mutated gene on to you, you too would get the disease. If mom has the disease, but she passes her healthy gene on, you will not get ALS. That's what happens in the autosomal dominant form. Some physicians make the mistake of asking you if your family has ever had ALS, and if you tell them no, then they don't test you because they think that you're not going to get it. A couple reasons that's a flawed reasoning. First of all, a family member could have died of heart disease or cancer or an auto accident or something before they got ALS in their 60s or 70s. You may not know their history. So that's not indicative of whether you have inherited a gene. It's also possible that they may have another mutation that impacts the expression of that gene. We'll hear about that in a few minutes. But it's also possible there are some forms of ALS that are autosomal recessive or simple recessive, meaning to get the disease, to have the phenotype, that you have to inherit the damaged mutated gene from both parents, one from dad and one from mom. In this case, if you have the recessive gene, your parents wouldn't have had this ALS. They wouldn't have shown the symptoms unless they had both copies of the mutated gene. And so that's another reason it's important to get your genotype, your whole genome sequenced. Now, there's another type of mode of inheritance where you only get the defective gene carried on your X chromosome. So your mom has 2 X chromosomes, your dad only has 1, and this can both be dominant and recessive. Very, very, very small percentage of people with ALS have this, but we wanted to give you an illustration here. If you have the mutation that is X-linked, you need to know if it's dominant or recessive. A lot of parents that have ALS ask me, is my child going to get this? And you won't know unless you know what your own mode of inheritance is. And ultimately, you may need to get your child tested as well. So just again, a summary, I'm not going to spend a lot of time on them, but wanted to give you the pictures of these, and you can certainly get back to any of us if you have questions about this. Now, familial ALS, we know that the most common form is now a mutation in your C9orf72 gene. The first gene that was discovered was SOD1, sometimes referred to as SOD1. And this one was discovered in the 90s. Now, depending on the type of mutation you have, that will impact your age of onset. The type of onset, whether it's limb onset or bulbar onset, may impact your speed of progression. But eventually with ALS, whether you have the familial form, regardless of what gene you have, or whether you have the sporadic environmentally caused form, you get similar pathology as this disease progresses. It impacts the nerves that innervate your muscles in your legs, your hands, your arms, your breathing muscles, and your mouth and tongue. We also know that there's some genes or things that can impact the expression of ALS. So for example, there's this protein SP110. If you have that and you have both copies of a SOD1 mutation, it will repress or suppress the expression of SOD1. So you will have the genotype, both genes for ALS, but you won't have the phenotype. They found that in canine research. Haven't explored that yet in humans. That's one of the areas we're hoping that somebody will take a look at for us in the human genome. We also know that if you have one type of gene that causes ALS and another type that causes a different form of ALS, those two also could combine. This is brand new emerging research. So for example, if you have the SOD1 gene and a copy of the C9 gene, and they're both recessive, but you happen to have one of each, there's sometimes ways that those genes can be impacting each other. And then we also know that some genes can have different mutations. You'll see that in a second. So this is just an example of the SOD1 gene and when it was discovered, and it's why there's so much more research about SOD1, because it's been around for almost 30 years now since they've known about it. And then this is when C9 was discovered, and you can see much later, which is why there's a little bit less research about it. Now, the other thing about genetic mutations is they can cause not only ALS, but they can cause other motor neuron diseases. So you can see from this study back in 2006 that some of these mutations in the spastin gene, causes damage to your upper motor neurons and can be related to ALS. You see this SETX gene, again, ALS, or can cause things like SMA and hereditary motor neuropathy. So very, very important to get your whole genome sequenced. We also mentioned that different genes can have multiple mutations, different places where there are mutations along that chromosome. So here in the SOD1 gene, the one we know about the longest, there are over 150 different types of mutations, and each of those mutations may have a different mode of inheritance. Most of them are autosomal dominant, meaning only from one parent, but some are autosomal recessive, meaning you have to get a copy of that mutation from both parents. We know of the people who have the familial form of ALS that get it inherited, about 20% of those cases are SOD1 mutations. We also know that one of the things that's increasing in prevalence is this mutation in TARDBP that causes problems with the TDP43 protein. Interestingly, that protein gets damaged both with the environmental and the genetic form of ALS, but this gene has over 40 mutations that they've found. Similarly, C9, back at the time that the C9 mutation was found, this study was from 2016, they thought it was only about 25% of cases. We actually know that that's closer to 40%. Just in the last 4 years, they found a lot more cases of that. Now, genetics can differ depending on what your ethnicity is and where you inherited those genes from. So if you are of European ethnicity, you are much more likely to have the C9 mutation, almost 15 times more likely than the Asian population. C9 had its origin in Finland, and that's why we see a large prevalence of ALS in Finland. It's also why we see a higher prevalence of the C9 mutation in the upper Midwest in the United States, around Minnesota and the Great Lakes, is because that's where a lot of people from the Norwegian countries immigrated here to the United States. In the Asian community, this SOD1 mutation is 13 times more likely than in the European community. And as you can see, it's very, very small incidence of SOD1 in the European community compared to the Asian community. Now, you're probably wondering, why do they show these genes being related to sporadic forms of ALS, the environmentally caused? Because what we found, researchers have found, is that sometimes the environmental exposures combine with the genetic causes, and together they create the phenotype or the symptoms of ALS, where alone one or the other may not. And again, new emerging area of research. We also know that ALS can affect children. It's commonly a disease that occurs more often with aging, but if it happens before the age of 25, it's called juvenile onset ALS. And you can see some of the different mutations that cause juvenile onset. Each of those mutations also, in a different manifestation, can cause the adult onset form, but those are the mutations that you would want to check for in your children if they start showing symptoms of this. Now, as we talk about ALS tonight, you're going to hear over and over things like oxidative stress, inflammation, mitochondrial dysfunction, glutamate excitotoxicity, protein misfolding and aggregation, degradation. All of the things you see here in blue are what we call the pathology, the things you're going to notice about ALS inside your cells, what's happening inside your cells. And each of these genes on the outside impacts each of these things. So the SOD1 gene, it controls your body's pharmacy that produces antioxidants. Most people don't know that our body has its own antioxidants. SOD1 controls that. And so the interesting thing and important thing for you to know is there are things you can do to impact oxidative stress or mitochondrial dysfunction, things you can do to help get rid of damaged misfolded proteins. And so regardless of which gene you have, almost everybody with ALS has all of these similar pathologies, maybe in differing amounts, but it's also a way that you can take empowering your body to tackle some of the things that are going on with it. One of the other reasons we want to suggest to you to get your whole genome sequenced is this is an example. This is a gene called HFE. It is not a gene that causes ALS, not one of those 24 plus genes, but HFE controls your body's ability to metabolize and handle iron. And we know that in people with ALS, they have high dysregulated iron levels. And so 30% of people with ALS have this mutation. And although it doesn't cause ALS, it will speed up the progression of ALS, and you need to be able to control or regulate your iron. So again, one of the reasons it's important to get your whole genome sequenced. This is another example. So you see here, these are all genes that the HLA gene over here, you see these genes are typically genes affecting immune function, but these genes also can impact your body's ability to handle those toxins, whether it's a virus or bacteria, whether it's pollutants, any of the toxins that are cause damage to you. And you can see examples of in PALS in a cohort is a group of people. So in a group of people that had ALS in Boston and another cohort in Israel, they found that there was a higher incidence of mutations in this HLA gene. You can see similar mutations found in Scottish cohorts, Finnish cohorts in the New York area. And here's what we also know is 25% of the population in general has a mutation in this gene called HLA-DR. So it's a really common mutation. That mutation impacts your body's ability to deal with toxins that cause systemic inflammation, like mold and fungi, viruses and bacteria, tick-borne illnesses, gluten, all of those things. Again, inflammation can lead to damage to your neurons as well. So these mutations in the HLA-DRA genes, DRB genes, they don't cause ALS, but they could leave you more susceptible to sporadic ALS. So it's really important to have your entire genome sequenced so that you know if you have any of these genes. You know, another thing that's really important, on the left-hand side, you see one of the leading ALS researchers who unfortunately just passed away of ALS just recently. His name was Rahul, and Rahul was studying polygenic. So polygenic means more than one gene, right? So again, he's looking at, and the people that are still working at his clinic are doing some of this genetic researchers talking about how different genes combine, how those mutations in different genes can cause you to have ALS, or perhaps just to lead you to be more susceptible to a rapid progression. But this polygenic factors, these are also other reasons that you want to have your entire genome sequenced. Now, sporadic ALS. We know that sporadic ALS can be associated with the genetic form, but now we're going to talk about the toxins and the environmental factors that impact you. So diet, viruses, bacteria, toxins from industrial pollutants and chemicals, things that you may work with through in your job, physical trauma like concussions, all of these things are different environmental factors. Now, on the right-hand side, you see a map, and this is the prevalence of ALS across the United States. For those of you that are curious, the black and the white dots that you see are where there are ALS centers of excellence or muscular dystrophy centers of excellence. The clinicians at those places are going to have a lot more expertise in dealing with ALS and diagnosing it. But if you look at the pink dots on the map, this is where you see the cases of ALS. And notice there's some areas with really high distributions of it. Michigan actually is the state with the highest prevalence of ALS. Ohio is right after that. And they believe that some of the reason may be because of the agriculture there, the pesticides, the herbicides, the fact that you have a lot of industrial chemicals and pollutants in the manufacturing that goes on up in those states. But you're also going to see incidents of high use of well water, which may be some of the causes both in the New England area and around the coast, where you can see a lot of states have a lot more incidents when they are right next to the coast. So we wanted to give you a couple of examples of where you may find what are called ALS clusters, and encourage you to know, when you talk to your clinicians, give them your entire history of where you've lived. If you grew up on a place that had what's called a Superfund site. Superfund sites is where the EPA has designated a site that's had a lot of pollutants that have been poured into the ground, the water supply there. One of the examples of that is there are two women that grew up across the street from each other in Kalamazoo, Michigan. There's a third person they went to school with the same age. All of them that grew up on this very same street all have ALS right now. They got it at different times, and it's impacting them differently, but all three of them have ALS. If you go and look at Kalamazoo, Michigan, where they're from, there were 5 or 6 different Superfund sites in their area. And importantly, they were polluting the environment from paper plants with PCBs. Is that what caused their ALS? We don't know for sure, but certainly if you come from a place where there's a Superfund site, you want your clinician to know what that was, especially if you're working with a functional medicine doctor. Here in Jefferson County, Michigan, there was another cluster. What was going on in Jefferson County? There was a lead smeltering plant right near them. There's, again, clusters in the upper Midwest and around areas of Great Lakes or oceans and bodies of water. Now, we know that working around agriculture, that you're getting exposed to pesticides and herbicides, and we're seeing high levels of those contaminants in people with ALS. We also know that in the military, your incidence of getting ALS is heightened, it's doubled. But in the people that served in the Iraq War, it's actually tripled, and they weren't really sure why. There's a couple of hypotheses, and all of these things we're talking about with the environmental exposures, we're going to be talking about hypotheses. There's not been any conclusive cohort studies about them yet. It's one of the things we want to try to help accomplish here with Everything ALS. But these are some hypotheses that have some pretty probative information. So this study was just came out here in 2020, and they talked about this pesticide that was sprayed on the uniforms of people that served in the military, and that pesticide may have caused problems when it was at high temperatures like you would have experienced during the Iraq War. We certainly know that landscapers, anybody that works with any type of farming or agriculture, you've likely had exposure to this. Anybody that works in the veterinary community where you may be giving flea and tick products to a lot of pets, and certainly some of the animals that come down with ALS have also had similar neurotoxic exposure from some of those pesticides. We talked about well water earlier. Here on the left, you see the place where there's higher incidence of people using well water, and there's been an association between well water and ALS. Why is that? Well water may have heavy metals in it. It may, because they're typically in agricultural communities, you may have more herbicides and pesticides that have contaminated the well water. There may be bacteria growing in there, but especially BMAA, which we'll talk about in a second, that comes from blue-green algae. So if you have well water and you have ALS, would encourage you to drink a healthier type of bottled water or purified water you can get your hands on. People with ALS, we encourage you to look at your amino acid levels and have your clinician measure these on a regular basis, because you're often deficient in some and higher in others. The essential amino acids are here on the left in the light blue. The ones that you see in bold are the ones that we typically see dysregulation in. And these have to come from food, your essential amino acids. And people typically think you don't need to get these from food, but people that are chronically ill will need to supplement if they're deficient in these on the right-hand side. And so what we know is, looking at some of these studies about amino acids, Dr. Bedlack, who's studying the reversals, he has not been able to conclude that it's causation, but he has found an association that 3 of the 45 confirmed reversals all have been taking high levels of amino acids. We know that when you study the amino acids in people with ALS, that there's typically some decreases in tyrosine and leucine, histidine. You may find increases in glutamate and cysteine. And we also find some dysregulated levels in L-serine. So why is L-serine important? So here you see a lot of different studies talking about L-serine, and that L-serine, if you supplement with that, it may be helpful if you have certain types of ALS, not all types, but certain types. So what you see here on the screen is a picture of why L-serine might be important. So remember earlier when we talked about well water, we mentioned the fact that well water may be associated with, excuse me after the coughing, we know that well water can be associated with the cyanobacteria called BMAA, but that's not the only place you find it. Initially, they discovered BMAA when they were studying this cohort, this population of people in Guam who had a higher incidence of ALS. In fact, it was 100 times higher than in the traditional population. And they ultimately determined that the BMAA was coming from this fruit that the natives were eating. And they ate this fruit, but they also made it into a flour that they used in their cooking. When they eliminated the fruit from their diet, their BMAA levels and the ALS levels went down a little bit, but not completely. What they found is this bat, called a flying fox, was also eating the fruit, and this bat was part of the native diet. So the bat was able to somehow tolerate these levels of BMAA without it causing neurotoxic harm to the bat, but it was resting in the meat and the fat and the muscles of the bat that the natives were eating. When they eliminated the bat from their diet, their incidence and prevalence of ALS decreased to the normal population as well. But BMAA is not just found in bats, it's found in water. So well water is one source. We also know that it's found in, it could be in the lakes that you're swimming in, could be in the ocean. So BMAA is, again, it's a cyanobacteria and it's a neurotoxin. When whales and dolphins have been washing up on the shore in the Gulf, they found that they basically have ALS, that they're having a neurotoxic effect from this BMAA. They found it in spirulina, really important because I know a lot of people take spirulina. It's a wonderful supplement that can help with neuronal repair, but you need to make sure you're getting it from a source that tests for BMAA to make sure it's BMAA free. But the other place that they have found it, in addition to being around the Great Lakes and any type of lake where the temperature of the earth is warming up the temperature of the water, and we're finding a higher amount of BMAA in our water, but they've also found it over in the Arabian desert where the Iraq War took place. So here you see them digging and they found BMAA in the sand and the soil. Between that and the insecticides that are sprayed on the uniforms, is that why we have a higher incidence of ALS in Iraq War veterans? We're not sure, but it's something that researchers are exploring. Now, we know that it's not just the bacteria found like BMAA, but just regular black mold. People that have a higher incidence of mold tend to be found more in the ALS population. So make sure you get a functional medicine doctor to assess your levels of mold, because mold and fungi can actually be treated with various different fungicides, depending on the type that you have. Now, this is one of the most controversial areas of ALS, is whether Lyme disease or tick-borne diseases are associated with ALS. Most researchers say no. Some of the Lyme researchers say no, but Dr. Schell is one of the functional medicine doctors that we work with here at Everything ALS, and her husband, unfortunately, has ALS as well. And she has found that people with ALS are 1,000 times more likely to have had Lyme disease. Now, we don't know what the association is. We know that Lyme disease can cause neurotoxic symptoms that are an ALS mimic. That could be one of the things. But does it cause long-term damage? Again, the researchers don't know. Most researchers say no. Interestingly, Dr. Merritt Kutkovich and Dr. Bedlack both did a study that was published back in 2009. You see this here. And what they did is they reviewed the medical records of patients that started back in 1989, going all the way up to when their study was published. And in that, they tried to see if Lyme disease was more prevalent in the ALS population. And they did not find that. They found that it really wasn't. Now, what's interesting, though, is that the tests for Lyme disease were a lot less sensitive back in the 90s and 2000s than they are right now. Functional medicine doctors and Lyme specialists are now doing a test called the ELI Spot assay, and a couple of other tests that are more specific to help detect Lyme or other tick-borne viruses in your body. So this Borrelia bacteria, this is what is Lyme disease, but some of the other tick-borne viruses, the people that believe that it is associated with ALS think what may be happening is that you may have the initial disease, that this bacteria somehow gets through your blood-brain barrier and gets into your neurological system. So even after you are treated and you seem to have recovered from the symptoms of Lyme, that there may be some ongoing inflammation going on inside your spinal cord because it passed through your blood-brain barrier. And we'll talk about your blood-brain barrier in just a few minutes. Now, we also know that viruses have a huge impact on people with ALS. Everything from polio, enteroviruses, retroviruses, HSV, so herpes simplex, HPV, HIV. We've even found a higher prevalence of some in the older population of people that have had measles and mumps, which are also RNA-based viruses. And one of the researchers is going to be looking at that for us. But we know when you have viruses, certainly this is all the talk right now with COVID, is viruses impact a lot of things. They cause inflammation. We've all heard that. They cause oxidative stress in your cells and calcium mishandling. They cause damage to your mitochondria. Again, damage to your RNA metabolism, and they cause protein misfolding. All of those same things, those are all the same pathologies you see associated with ALS. And so it's one of the reasons we want people to, again, pay more attention to, talk to your clinician about every single virus you can remember having. Could be very important in helping diagnose what some of the causes or risk factors may have been for you. We also know, and this is critical, so gut health plays a big role in ALS. What we don't know is why. So we know that gut health, or the good bacteria in your stomach and your intestines, that it's not the cause of ALS, but it's somehow playing a role with ALS. We don't know if it's the genetics that are triggering problems in your gut microbiome, or is it your gut microbiome that's allowing damaged toxins to get into your blood past the blood-brain barrier. Not sure about that. But one of the things we want to mention is, for PALS, make sure you get that fecal sample done so you know what gut bacteria you're high in and what you're low in. You may need more bifido, less lactobacillus. Most people with that we have seen anecdotally that we've talked to that are PALS tend to have problems with their lactobacillus. Another thing that they're often low in is this Akkermansia. And Akkermansia you get from pomegranates, other fruits. And it's really important to make this metabolite called nicotinamide that's like vitamin B3 in your body, very important for your body's function. So make sure whatever probiotics you're taking, that they don't get eaten up here by your stomach acid. Almost all probiotics are wasted here and your stomach acid destroys them. As you can see, you have a lot more bacteria here in your intestines, and this is where you want to get these good gut bacteria to be. And if they get eaten up here in your stomach, it's not going to help. So make sure that your probiotics survive that. That means they have to either be spore probiotics or they have to have a protective coating on them. Liquid probiotics typically don't make it down into your intestines. So make sure you're talking to your clinician or your functional medicine practitioner about the best type of probiotic for you to use based on your own specific fecal sample. So probiotics are not going to be one size fit all, and you need to adjust your probiotics based on your own gut microbiome. So this is an example of what happens when your microbiome is not working well. These are called tight junctions. So see how these cells are really close together here, and only the good things, the good vitamins, are going to be able to get into your bloodstream. When these tight junctions are broken down, you can see viruses, bacteria, pollutants, toxins, all of these unhealthy things can get through your stomach lining because they're no longer tight junctions, and they get into your bloodstream. And once they're in your bloodstream, some people believe that's how they get into your neurological system, because they pass the blood-brain barrier. Again, new emerging area they're looking at. But one of the things that your clinician can test for that's so important is this thing up here in the left called zonulin. Starts with a Z, Z-O-N-U-L-I-N. When you have breakdowns in your tight junctions, you're going to have a higher level, you're going to note a change in your zonulin levels. So make sure that your doctor is testing your zonulin levels. And then, as you can see here, you see the toxins getting into the bloodstream, and then this is your blood-brain barrier. And again, if some of these toxins may be getting in to damage your brain and your nervous system, excuse me, and what that causes is inflammation, an immune response, free radicals, that's oxidative stress, glutamatic cytotoxicity, all of these things, again, related to ALS, and that could be contributing, the problems with your gut health can be contributing to that. Now, one of the other areas that is emerging is the impact of concussions on ALS. So we know that concussions, there's a whole lot of people outside the NFL or rugby that have had them, people that are biking, maybe you have a skiing accident, but about 20% of high school kids have had at least one concussion. So this is a big deal to pay attention to. And we believe this may be one of the reasons that men have higher incidence of ALS when they're in a younger age group. So we know that if you have had a concussion, what happens is you get oxidative stress in your brain, and then you also get neuroinflammation or inflammation inside your neurons and inside your brain. And when that happens, it leaves you more susceptible to Parkinson's, to ALS, to Alzheimer's, and to dementia. And so you may also have heard of some of the people in the NFL having CTE, chronic traumatic encephalopathy. So this may also be, some people have said this may be why some of the soldiers have a higher incidence of ALS, because of shooting high rounds of artillery or having concussions when they're training, right? Now, they did a study of some professional athletes, people in the NFL, professional rugby players, professional soccer players, along with a few other sports where they didn't typically have concussions. And what they found is those sports, when there was repetitive head and cervical spine trauma, so head and neck trauma, that the risk of ALS was 850% increased. In the top, you see a picture of Dwight Clark, who unfortunately passed from ALS. He played for the Niners. You see on the screen here in the red, all of the people that are professional athletes who have had ALS or we've lost to ALS. You saw Tim Green earlier on the screen. We know Steve Gleason, Bob Waters, Pete Frates, Catfish Hunter, a baseball player, Orlando Thomas, right? A lot of professional athletes and collegiate level football players, soccer players, rugby players who have had repeated concussions. This can be an issue. Could also be somebody who participates in the rodeo, somebody who's breaking horses, anybody that's been thrown from a horse several times. Any of those high impacts on your head and neck could have repeated auto accidents that could also contribute to this, that can lead to an increased incidence of ALS. Now, we also know that heavy metals may play a role, and this is again a controversial area. So alone, researchers don't believe heavy metals cause ALS, but they're certainly in conjunction with several different other risk factors. They may be playing a role. And so we wanted to show you where you're going to see some of those exposures. So heavy metals, everybody's heard about the lead in the water supply in some of the older cities on the East Coast, like Flint, Michigan, Newark, New Jersey. If your city has older lead pipes, you're going to have more lead in your water supply. We've all tried to be cautious about lead paint. We know that you get higher levels of mercury the bigger the fish that you eat. So swordfish and Chilean sea bass have higher levels of mercury than a smaller fish like halibut. We also know that the amalgam fillings in your mouth can have a lot of damage. If you have amalgam fillings, we recommend you contact an integrative dentist, because taking them out can cause even more harm than having them in, and you need to make sure they're removed properly. You also need to make sure you're working with a functional medicine doctor, because you may need to do chelation at different times depending on the types of metals you have in your body. You see, if you work with welding, if you've done any steel erection and you've been working with welding, you may be inhaling those metal fumes. Women, all of us, unless you're using 100% organic products, our cosmetics, our lipsticks, our blushes, they are going to have heavy metals in them. They did a test of baby food, and several different brands of baby food had levels of heavy metal in them. You see on the right, they tested 45 different types of fruit juices, and they had high levels of lead, arsenic, and cadmium found in every single one of them. Obviously, you can get heavy metal exposure from the pollutants in the air, depending if you're working around a coal plant, if you're around a lead smeltering plant, any type of industrial work where you're around those pollutants. Heavy metals are in our pollution as well. So we know from some of these studies that there's different levels of heavy metal that have been found all around the world in people that have mercury, in people that are in Spain, in France, in the United States, and it may vary depending on your area. And again, these can also be found in your well water, depending where you live. And they found a higher risk of ALS, slightly higher risk, if you have lead, arsenic, cadmium, mercury, zinc, chromium, and copper. Interestingly, copper is related to SOD1, and you may need zinc to balance out that copper. So that's another reason it's important to have your zinc and copper levels tested, but also why it's important to have your genome sequenced. What we also learned just this year, in May of 2020, they released a study about the levels of heavy metals that are found in people's teeth. So your teeth have rings in them, just like the rings in a tree. And the researchers called them a biologic time capsule, said it's like a hard drive for your body, knowing what you were exposed to. Again, you can see in the people with ALS, they had higher levels of chromium, manganese, nickel, tin, and zinc. We've also found people, not in this study, but anecdotally, a lot of people that have dysregulation of aluminum as well. So again, make sure heavy metal levels are tested and you work with a functional medicine doctor on that. We also know that one of the other environmental associations that we've found is with smokers. So 42% more likely to be diagnosed with ALS if you are a smoker. The risk of ALS increases the younger you started smoking. And for every 10 cigarettes you smoked a day, your risk of ALS increases 10%. And unfortunately, there's a younger age of onset if you're a smoker. And the bad news, unfortunately, doesn't stop there. The speed of progression of ALS is quicker. It shortens, cigarette smoking shortens your survival time if you have ALS. And researchers are hypothesizing that that could be because of the chemicals and pesticides that are in the tobacco. It could be the nitrous oxide and chemicals that are in cigarette smoke, but also heavy metals, again, like lead, nickel, cadmium, and chromium. Now, these are some of the occupations that have a higher risk of ALS. And some of this came from a study at Michigan, where they've really been a leader looking at some of the OSHA data here in the United States. So you can see if you've been in the military, 2 times increased risk, 3 times if you were over in the Iraq War. Again, could be from the concussions that you've experienced, could be from some of the exposures to herbicides and pesticides, the industrial pollutants that you're using, or some of the petrochemicals you're exposed to around, around the equipment, the tanks, the aircraft. For pro athletes, again, 850% increased risk if you have had concussions. It does not seem to be an increased risk if you are in a sport where you are not having concussions. Firefighters have an increased risk because of the chemicals in the fires that they're putting out, industrial fires, but also importantly, the fire retardants that we use on our carpet, on our furniture, on our clothing. Those fire retardants are associated with ALS. Landscapers, farmers, anybody that works with agriculture, you have a higher incidence of ALS, likely because of the herbicides and pesticides, but also perhaps because some of the well water. Anybody that deals, painters or laborers, because you're dealing with industrial chemicals. Anybody, again, steel erectors, people that deal with heavy metal exposures, people in the mining industry have a lot of that, steel erection industry. Miners are also exposed to the wastewater and the things they're dealing with, the pollutants that come off of the mining process. If you work in construction, if you're a truck driver, if you operate heavy equipment, again, diesel fuel, jet fuel, people that are working with petrochemicals, a higher incidence of ALS. And then if you're working on an airfield where you're constantly being exposed to radar and EMF at those high levels, they've seen a higher incidence of ALS in that community as well. Now, we're going to talk briefly about the pathology of ALS. We've heard us mention some of these terms about oxidative stress and mitochondria and protein, and we wanted to give you an idea of what they were. So these are all of the different things that can be impacted at the cellular level when you have ALS. And these are some changes that are probably going on at the cellular level before you ever see the symptoms. And it doesn't matter if you have the environmental exposure, the sporadic ALS, or the inherited form of familial ALS. So this is a picture of a neuron. You can see here, this is mitochondria that's damaged. And anytime you see ROS, that's basically oxidative stress that's going on in your body. You see damage to your DNA. We see oligodendrocytes and microglia that have damage, protein misfolding. And again, you're seeing some of the genes here that are associated with each of those things. Down here, microglia damage. Here you have transport and cell signaling problems, glutamate excitotoxicity, oxidative stress that comes from SOD1, TDP43, and C9, all three of those, the most common forms of genetic ALS, and then even mitochondrial damage. So we mention these over and over, but a lot of people don't know what they are. So oxidative stress it's not stress like most people think about. It's actually what happens inside your cells when you have less antioxidants than you do toxins or free radicals. And so this is actually something we can impact, right? We can boost both our body's production of its own antioxidants, and we can take antioxidant supplements. So what is an antioxidant and how does this free radical damage it? So we wanted to give you an illustration. If you remember back to your biology classes, we know that a molecule has to have an equal number of electrons to be balanced and stable. A free radical is just missing an electron. And so what happens is this free radical, it's damaged and it bounces around inside your cells, causing damage and destruction wherever it goes, right? Kind of like leaving footprints when you come in the house with muddy feet. It's going to leave damage wherever it goes. The way an antioxidant works is it takes this extra electron and it gives it to the free radical and balances that free radical. And then you want to get it out of your cells. Now, here's what happens with people with ALS or anybody who has oxidative stress. You want to try to deal with it at the cellular level here, because if you don't, it's going to expand and cause damage to your tissues, to your organ, and ultimately to your entire organ system. In ALS, oxidative stress is causing damage to our entire neurological system. So we need to do a lot to lower that oxidative stress levels and hopefully to be able to eliminate some of the damage that's happened at the neuronal level, at the neurological level, right? Now, there's different types of antioxidants, and we wanted to go over these with you. So the ones you see in the light blue at the bottom are the supplements you take. These are called exogenous, EX for external to your body. And you get these from your diet, blueberries, fruits, vegetables. You can get them from supplements, vitamin A, C, and E supplements, CoQ10, glutathione. Those are all powerful antioxidants. But the antioxidants in the top are the ones that your body's own pharmacy makes. So most people don't know that your body makes its own antioxidants. And these three at the top are way more powerful than the antioxidants you see at the bottom. So the glutathione is called the master antioxidant, not only because of how powerful it is, but also because it makes the other antioxidants more powerful. And glutathione peroxidase your body makes. Catalase is another antioxidant that your body makes. And then interestingly, SOD, SOD. So the SOD protein is important to turn on your body's antioxidant functions. There's other genes that also turn on your body's antioxidant function. You want to upregulate those genes. Remember, we talked earlier about epigenetics. You want to turn on the genes that turn up your antioxidant function to help your body get rid of those toxins and free radicals. The higher you go up this hierarchy, the more powerful these antioxidants are. So we also know that inside our body, we have about 130 sextillion free radicals. People with ALS likely have even more than that. And if you just think about our neurological system, there's billions of neurons in our brain, in our neurological system. So if each of these neurons has oxidative stress in them, it's really important that we decrease those levels of oxidative stress. Let's talk mitochondria. So this is a picture of a cell kind of cut in half. And what you see here, this is the nucleus of the cell, and these little things that look like worms, those are mitochondria, mitochondrion singular or mitochondria plural. And mitochondria are like the engines of our cells, and therefore the engines of our bodies. They are what gives our heart the energy to beat, our brain the energy to think, our muscles the energy to move. And so mitochondria give us up to 95% of our body's energy. And whether you have ALS or not, your quality of life is going to depend on your quality of mitochondria. A variety of different diseases, not just ALS, not just neurodegenerative diseases, cardiovascular diseases, a lot of different diseases, people have dysfunctional mitochondria. And so mitochondria are so critical to help us boost our function. Now, the picture's a little bit deceiving because it only shows 3 mitochondria. What we know is that in our bicep muscle, for example, there may be 250 mitochondria per cell, not just in the muscle. When we get to our neurological system and our cardiovascular system, there could be 5,000 or 10,000 or maybe even 100,000. The place that has the most mitochondria is in your eggs, your ovary, if you're a woman. So the more mitochondria you have per cell, the more likely you're to be impacted if your mitochondria is damaged. And so that's one of the reasons people with ALS focus so much on boosting their mitochondrial function. So on the left, you see a picture of a mitochondrion and some of the things that impact it. And we wanted to give you a way to think of mitochondria that's a little bit simpler. So we want you to compare it to logs on a fire. What do you do when you want a bigger fire, a healthier fire? You want more logs on the fire and you want big, thick, robust logs. Same thing happens in your cells. For your cells to function optimally, you want more mitochondria and you want big, thick mitochondria. Athletes like Michael Phelps, endurance athletes, they have more mitochondria. As you are immobile, your mitochondria die inside your cells. So important to keep our mitochondria boosted. But also go back to the fire analogy. What happens after you burn a fire? The waste product from the fire is soot at the bottom of your fireplace. And if you never clean the soot out of your fireplace, your fire's not going to burn as well. The same thing happens in your cells. The waste product from mitochondria producing energy is free radicals. So you get free radicals from the mitochondria, you get free radicals from the pollutants, from the toxins, from the bacteria. And you've got to find a way to get those free radicals out or you're going to have too much oxidative stress. And if you don't clean the free radicals out of your cells aren't going to work, just like if you don't clean the soot out of your fireplace. Now, these are just some studies that show you the correlation between neurological health and mitochondria. Very, very critical. You can see they talk about overwhelming evidence that mitochondrial function is one of the causative factors of neurodegenerative diseases. We mentioned before misfolded proteins. So we gave you an illustration on the top. This is what your SOD1 protein looks like, and this is what happens when it gets misfolded. So a lot of things can cause proteins to misfold. Again, can be genetics that cause it, or it can be environmental toxins that cause the damage. Think about a misfolded protein. Again, we'll go to a simpler analogy like a bunch of grapes that you would eat. And if one of those grapes has mold on it, and you don't remove the grape from the bunch of grapes, what happens? That mold spreads and damages the entire bunch of grapes. Same thing happens with damaged proteins inside your body. They get misfolded, and then if you don't remove that damaged protein, it's going to cause damage to other proteins, and then they're going to clump together, and they're going to misfold together, and those are going to cause, again, more damage to your neurons. So TDP43 is an example of that protein that often gets misfolded. The SOD1 protein misfolds. You see down here in this study, they talk about the different protein misfolding problems. Now, how do you get rid of it? How do you get rid of those damaged proteins? So one of the ways is with autophagy. So autophagy means self-eating. Think about a little Pac-Man eating the other Pac-Man. This drawing here, you want to get rid of the toxins and misfolded proteins and get them out of your body. And your autophagy is your body's recycling system. It's the way to get the junk out of your cells so that you can have new healthy cells that grow, right? And so there are, again, are things you can do to boost your body's autophagy and work with your functional medicine doctor on that. We mentioned glutamate excitotoxicity. We're not going to spend a lot of time on it because it's a little bit harder to alter, but you can see here, this is the normal level of neurotransmitters or glutamate that's going between your two neurons here. And then this is what happens when you have too much glutamate here and you see increased oxidative stress and increased calcium, right? So another area that we want to control. Now, one of the things, there's a great book called Lifespan by Dr. David Sinclair at Harvard. And in his book, he talks about the hallmarks of aging. And he says, listen, we used to believe that disease caused aging, but there's been a paradigm shift. We now know that aging is what leaves us susceptible to disease. And so if we can find a way to slow down the aging process, increase our longevity, we know that we can slow down disease and hopefully delay human death. And we find a big correlation with the increase of the incidence of ALS as we age. And you see that represented here on the graph. When we compare what happens to an aging neuron, and this is an aging neuron, and these are all the things that happen to it, along with what happens in an aging body of somebody who doesn't have ALS, you're going to find some similar things, unfortunately. So you're going to see mitochondrial dysfunction, an increase in oxidative stress. You're going to find a decrease in muscle mass and strength. In ALS, you're going to find motor neuron degeneration. That's a little bit unique, but you're going to find problems with inflammation, DNA damage, cellular death, protein aggregation. All of these things we've been talking about over and over and over again are going to happen to people that age along with people that have ALS. And so if we can try to get a handle on some of those things that are causing aging and turn on our genes that protect us, this is one of the genes that does that. It's called a sirtuin. And so when we talk about aging, there's 9 different hallmarks, and you will have heard these mentioned over and over again. And these are the things you can do to address those hallmarks of aging. A lot of them are what some of the people in Dr. Bedlack's reversal studies are focusing on some of these different things. One of the things that is easier for you to impact are your genes called sirtuins. So these are 7 genes that all mammals have. And these genes are called longevity genes because they found that people that lived to be 100 tended to have sirtuins that were working better. Unfortunately, by the time we hit middle age, for most people, their sirtuins are about 2/3 less effective, basically only functioning at about 1/3 of our capacity that they did when they were younger. And these genes control things like mitochondrial health, oxidative stress, neuronal regeneration, a whole lot of different functions that keep us young and vibrant and healthy. And they don't work as well as we age. Part of the reason is because of this molecule. This molecule is called NAD, and a lot of you may have heard of NAD. There's people that are using NR or NMN, vitamin B3, tryptophan, niacin, niacinamide. Those are all, nicotinic acid, are all precursors to NAD. And what we know is these sirtuin genes, all 7 of them, they need NAD to function. And as we age, we don't make as much NAD. And so researchers are really looking at how NAD is helping improve the sirtuin function, which is having an impact on ALS. There was a brand new study that just came out yesterday. Today is June 21st. Brand new study that just came out that talked about the impact of NAD and caffeine together that boost your NAD function as well. These are some of the different studies that show the correlation between NAD and sirtuins. So we want to boost those sirtuins. There's other genes that also can impact your body's aging process and oxidative stress. One of those is called NRF2 activation as well. Now, reversals. Let's talk about Dr. Bedlack's reversals. He's going to be speaking in a couple weeks on one of our presentations, so I'm not going to spend a lot of time on this, but just want to go over this if this is your first exposure to the fact that there are some people who have reversed their ALS. And what they've done is they've addressed again, some of these pathologies, they're decreasing their inflammation and oxidative stress, their mitochondria. Now, this is Dr. Richard Bedlack at Duke's Med School's ALS clinic. He is both a researcher and a clinician, and we are so grateful to him in the ALS community because he's the first person in the medical community that decided to study the people who had reversed their ALS. Now, he studies these AOTs called alternative off-label therapies. He's got two studies, a STAR study where he looks at the different people that have reversed and tries to see if there's any plausible mechanisms. He verifies that they are a valid reversal. He does that by looking at their records, making sure it's not an ALS mimic. He makes sure they have a significant improvement in their ALS FRS scores, and that must have occurred over at least a year's period of time. And some of his reversals have been for more than 10 or 20 years. These are some of the people that spoke at a conference last fall, in the fall of 2019, who have reversed their ALS. And Dr. Bedlack found some similarities amongst the people that he studied in his confirmed reversals. So at the time he published this study, he said the average age of his reversals was 50, but they ranged from 18 all the way up to 75. They were more likely to be male. 34 of the 43 were men. They were more likely to have limb onset, but there were 3 that had bulbar onset. There was an equal proportion of people with the genetic form versus the sporadic form. So 10% of his reversals had the genetic form, the other 90% had the sporadic form. And interestingly, most of them had a rapid progressing ALS before their reversal. But one of the things I also found interesting is that all of his reversals had re-innervation. That's what you see here on the screen. So remember our example from before, where we have these, looks like the roots of the tree going into the muscle. And when the neuron dies, these roots, they die as well. And so this is a drawing of this from a polio patient. So when you have healthy neurons, you have these roots extending into these little muscle fibers here. When you have a neuron that dies, those roots die into this muscle fiber. So this muscle fiber wasn't getting the signals here, these muscle fibers. What happens with reinnervation is the existing neurons, the ones that survived, they continue to grow new roots to help reinnervate, create new connections with those muscle fibers. You see other illustrations down here at the bottom. So this is a healthy neuron. You've got the axon terminals extending here into the muscles. When this right neuron on the side dies, this one here on the bottom, it no longer extends into this part of the muscle. And so that muscle is not going to get the signal to fire. And what happens with reinnervation is all of these reversals have had new sprouting from the healthy neuron that's remaining. So they don't know why that is. Researchers are looking into how they can support reinnervation as well, but a finding that is fascinating, and now we need to find out why it's happening and how we can repeat it in other pals. So these are 2 of the studies that Dr. Bedlack has studied with people that have reversals, which is less than 1% of the population. He's found some common supplements, glutathione, which again, a huge antioxidant, it's our master antioxidant. Curcumin, which is also an antioxidant, anti-inflammatory, another antioxidant, anti-inflammatory. Fish oils are super important for neuronal health, myelin sheath, but also anti-inflammatory. Vitamin D, copper, which is critical with SOD, cannabis, and an immunosuppressant. Now, he's very specific that he has not found causation. He cannot tell you that using these will reverse your ALS. What he said is he's found associations, meaning the people that had reversed were taking these substances, but he doesn't know if that's why they reversed. It may be because they had a special gene. It may be because they did chelation. We don't know the cause yet, just that there's an association. Now, you can look at his studies on a website called ALS Untangled, where he goes over clinical mechanisms, and he gives each of these supplements a grade from A to F on whether there's clinical possibility, whether there's any cases or any trials, and he looks at the risks as well. And then after he looks at those, he does a review. He and the other 200 researchers from around the world write up a review. So this is a completed review on a supplement called methylcobalamin, which is basically a bioavailable form of vitamin B12. And there's a really good write-up. He gave this an A rating. It's one of the few supplements that he's given an A rating to. There's also a place on ALS Untangled where you can look at other supplements that they haven't reviewed yet, and you can vote to have them review that supplement protocol or that individual supplement. Now, we put together a list of some of the supplements that Dr. Bedlak has given A ratings to, or the ones that he talked about in his studies, as well as some of the supplements that a lot of different PALs are taking, just to give you a list. We're not endorsing the use of these. We're just giving you an overview of what anecdotally has been, that if you are new to ALS, these are some of the most common supplements that people with ALS are adding to their protocol. We want everybody to make sure you work with a functional medicine doctor or a naturopath before you add these, and these should be based on your own blood work and your own blood test and your own type of ALS, because you may need more or less of each one of these depending upon the type of ALS and your own specific blood panels and urine panels. Now, these are some of the people. We just wanted to let you know that it is possible. It's less than 1% of the population, but these are some of the people today who have reversed their ALS. So this is Stephen Bishop. Up in the top left, he was in a wheelchair. He's now walking, golfing, and hiking. Eric Edney lived into his 80s. He was a long-term survivor. He wrote the book, Eric is Winning, so you can look at his protocol. Derek Swinard was a basketball coach up in Canada. He still golfs today. Mark Manchester was on a feeding tube and a trach, and he is now walking and holding his new grandbaby. This over here on the right is Kim Cherry, who had bulbar onset. Kim and his wife have done a lot with working with naturopaths, all sorts of different supplements. This is Gunther. Gunther reversed his ALS. We lost Gunther to a heart attack last year, but he had reversed his ALS and was skiing and hiking and bringing a lot of joy to the ALS community. This is McFinn, who was with us here at Everything ALS. He was down to just the use of 2 fingers, and that's it. And he now runs his own property. He plants, he walks, he hikes, and gives lots of inspiration to people in the ALS community. This is Stephen Sherry, Sherry. He lives down in Texas. Again, worked with a naturopath, does a high volume of antioxidants in his supplement protocol. He's a long-term survivor as well that has reversed his ALS. And this is Deanna. Her father was a doctor, an MD, and when she was diagnosed with ALS, he came up with the Deanna protocol, and you can look that up online if that's something you're interested in exploring with your clinician. And even if you look at Dr. Jocker's website, again, functional medicine, and you talk about what he believes supports your body with ALS is to do things that decrease inflammation, support your mitochondria, get rid of those toxins, chelate, balance your gut microbiome, heal your leaky gut issues, optimize your micronutrients like your vitamins and minerals, boost your glutathione, eat omega-3 fatty acids in your foods and maybe supplements, and maybe even some ozone therapy or hyperbaric chamber therapy. Saunas are a lot of people are using as well. But as we close with Dr. Bedlak's session here, we just want to mention, again, he is not saying that this is causation. He's just researching it, and he's found association thus far. We are so grateful for Dr. Bedlak, who has taken this as one of his missions to find what's helping these people reverse ALS. We hope you're able to join us at the end of July when he's going to be speaking, talking about that specifically and giving you a lot more detail. Now, with nutrition, we just want to encourage you, we'll bring in a nutritionist to talk to you a little bit more on one of our Wednesday webinars, but there's a book that might be really helpful to you, and a lot of PALs are following it. It's the Walls Protocol. It's written by an MD, Dr. Terry Walls, who has MS, and she was able to reverse her MS by her diet. So it's a fascinating book about eating organic, about lots and lots of vegetables and fruits that do things like boost your antioxidants and boost and support your mitochondria. So all of the things we've been talking about repeatedly tonight, she has reinforced with nutrition. Encourage you to get that. If you have family member that's using a feeding tube, a PEG, again, take a look at what's in the formula that's going into their feeding tube. A lot of them are junk. It's corn syrup and sugars and empty calories, and all of those things are harder on people with ALS. So try to find a formula for your feeding tube that is going to be healthy for you and help support your body's defenses. And as we close tonight, we just want to thank you for joining us here with Everything ALS. I'm just going to end this presentation. Thank you again for joining us with Everything ALS. We hope you join us every other Wednesday night and help you find both what we can do to support you from the care stage of ALS all the way to the cure. Thank you for joining us. Hope everybody has a really good evening.